Physiology SAQ9 explain the effectiveness of the following systems as buffers in the body. A. Bicarbonate carbon dioxide system, 5 marks. Introduction. A buffer is a substance or a mixture of substances that minimizes the change in pH of a solution that would otherwise occur when an acid or base is added to it. In aqueous solutions, this comprises either a weak acid and its conjugate base, or a weak base and its conjugate acid. For the equation, Ha becomes H plus A negative, where Ha is undissociated acid and A negative is the anion. The equation shifts to the left if acid is added and to the right if base is added. Changes in H plus concentration are thus minimized. The Henderson-Hasselbalch equation describes these relationships. pH equals pKa plus log A negative divided by Ha. When pH equals pKa of a closed buffer system, maximal buffering occurs because Ha and A negative exist in equal amounts. Buffers within body fluid stabilizes the pH by binding to excess H plus or by releasing H plus. Three important buffer systems function together to prevent major changes in pH of body fluids: the carbonic acid bicarbonate buffer system, the protein buffer system such as hemoglobin and plasma proteins, and the phosphate buffer system. The carbonic acid bicarbonate buffer system. H2CO3 or carbonic acid is a weak acid formed when CO2 and water mix. When it is dissolved in water, the following equilibrium is established. CO2 plus H2O becomes H2CO3 in the presence of carbonic anhydrase, which equilibrates with HCO3 and hydrogen ion. When H plus is added to this solution, a large proportion of the H plus binds to HCO3 negative to form H2CO3, and only a small percentage remains as free hydrogen ions. Thus, the carbonic acid bicarbonate buffer system resists a large decrease in pH when acidic substances are added to a solution containing H2CO3. When H plus is removed from a solution containing H2CO3, much of the H2CO3 forms bicarbonate and hydrogen ions. Thus, a large increase in pH is resisted when basic substances are added to a solution containing carbonic acid. In general, an effective buffer system has a huge capacity or high reserve, rapid in action, straddles more than one organ system so that if one organ fails, the other may be able to assist in compensation, i.e. open system. And the buffer system has a pKa value close to physiological pH. The bicarbonate carbon dioxide system is effective as bicarbonate is in abundance, it has rapid action, and it is an open buffer system involving the respiratory and renal system. Despite the pKa of bicarbonate system being 6.1, its effectiveness as a buffer is greatly increased because the system is open at both ends. This means that both the PCO2 and bicarbonate ions can be altered, and this assists in minimizing changes in the bicarbonate to PCO2 ratio, termed in the Henderson-Hasselbalch equation. The excretion of CO2 by the lungs to maintain a constant PaCO2 greatly increases the effectiveness of the system as a buffer, especially as changes in CO2 excretion can occur rapidly. Bicarbonate excretion can be regulated by the kidneys. When hydrogen ions decreases, the distal convoluted tubules decrease H plus secretion into the urine and decrease bicarbonate reabsorption into the blood. When H plus increases, the DCT increases hydrogen ion secretion into the urine and increases bicarbonate reabsorption into the blood. B. Hemoglobin, 5 marks. Introduction to the protein buffer system. Intracellular proteins and plasma proteins forms a large pool of buffer molecules. Hemoglobin in red blood cells is one of the most important intracellular proteins. The capacity of proteins to function as buffers is due to the functional groups of amino acids such as the carboxyl COOH or amino NH2 groups, which can act as weak acids and bases. Consequently, as the hydrogen concentration increases, more hydrogen ions bind to the functional group, When H plus concentration decreases, hydrogen ions is released from functional groups. Hemoglobin as an effective buffer depends on hemoglobin's abundance, its rapid action, hemoglobin being a weaker acid than carbonic acid. The pKa value is close to physiological pH. Deoxyhemoglobin being a more powerful buffer than oxyhemoglobin. The important role of imidazole groups of histidine residues in hemoglobin and carbaminohemoglobin formation. Hemoglobin's abundance. Hemoglobin is about 6 times more important quantitatively as a buffer compared to plasma proteins, as hemoglobin is twice as much versus plasma proteins, 15 grams per deciliter versus 7 grams per deciliter. Hemoglobin has a rapid action as a buffer. Hemoglobin is a weaker acid with a pKa of 6.8 compared to that of carbonic acid with a pKa of 6.1. Hemoglobin exists within the red cell as a weak acid HHB and its potassium salt KHB. As a result, H+ is buffered by hemoglobin and bicarbonate is increased proportionately as shown in the following reactions. As a result of these reactions, red cell bicarbonate concentration increases and the bicarbonate diffuses out of the red blood cell into the plasma to maintain electrical neutrality. The pKa value of hemoglobin is close to physiological pH. Buffering capacity is maximal at the pKa of the weak acid for the majority of the buffer system. Majority of buffering occurs at pKa plus minus 1 for closed buffer systems. Deoxyhemoglobin being a more powerful buffer than oxyhemoglobin. Deoxygenated hemoglobin has a higher affinity for CO2. Thus, oxygen unloading assists in increasing the carriage of CO2 by hemoglobin due to the Haldane effect. The Haldane effect causes upward shift in CO2 dissociation curve. 30% of the Haldane effect is due to deoxyhemoglobin being more basic and therefore able to buffer hydrogen ions more effectively than oxyhemoglobin. CO2 dissolves into RBC from tissues. Carbonic anhydrase is present in RBCs. CO2 plus H2O produces H2CO3, which dissociates to hydrogen ions and bicarbonate ions. Bicarbonate dissolves out of the RBC with chloride inward shift, the hamburger effect. Hydrogen ions are buffered by imidazole moieties on histidine residues of the globin chain of hemoglobin. The hamburger effect and hydrogen ion buffering by hemoglobin maintains the shift of this equilibrium to the right, and CO2 is effectively transported as bicarbonate. With an arterial transport of CO2 of 90% bicarbonate and AV difference of 60% bicarbonate. 70% of the Haldane effect is due to deoxyhemoglobin being more effective at forming carbamino compounds than oxyhemoglobin. CO2 diffuses into RBCs to form carbamino compounds. For arterial transport of CO2, 5% is transported as carbamino compounds, and the AV difference is 30%. Deoxygenated hemoglobin has a pKa of 8.2 and dissociates more than oxyhemoglobin with a pKa of 6.6. At the normal blood pH of 7.4, both forms of hemoglobin are equidistant from their pKa values, and so both are theoretically equally effective buffers. However, oxyhemoglobin is able to resist a decrease in pH, whereas deoxyhemoglobin is a better buffer when pH tends to rise. Therefore, in tissue capillaries, oxyhemoglobin releases oxygen and is reduced to deoxyhemoglobin, facilitating the uptake and buffering of hydrogen ions generated by the hydration of CO2 and dissociation of H2CO3. Isohydric buffering. For each millimole oxyhemoglobin that is reduced, 0.7 millimole of hydrogen ion can be taken up and 0.7 millimole of CO2 can enter the venous system without changing pH. The advantage is that in capillaries, after oxygen unloading, oxyhemoglobin is reduced and has an increased buffering capacity. This explains why pH of venous blood is only slightly more acidic than arterial blood. Role of imidazole groups of histidine residues in hemoglobin. Each hemoglobin contains about 3 times more histidine residues than the average plasma protein at 38. An imidazole group of histidine is the only amino acid residue which provides buffering at physiological pH at its average pKa of 6.8. Carbaminohemoglobin formation. HbNH2 plus CO2 produces HbNH COO negative plus hydrogen ions. HbNH2 plus H2CO3 produces HbNH3 plus bicarbonate ion. CO2 and H2CO3 binds with amino acid groups, forming carbamino compounds, which accounts for 15% of CO2 transport. Additional information. The phosphate buffer system. Phosphoric acid is a tribasic acid. At physiological pH, the biphosphate H2PO4 buffer is the main phosphate buffer. When pH decreases, ions such as HPO4 -2 binds hydrogen ions to form H2PO4 negative. However, when the pH becomes more basic, H2PO4 negative releases hydrogen ions into the solution. In this way, these two ions fluctuate between gaining and losing hydrogen ions, which helps balance the pH. On a purely chemical basis, it is potentially the most efficient buffer in the extracellular fluid, as its pKa is 6.8. As plasma phosphate concentration is low, 0.8 to 1.4 millimole per liter, and phosphate operates as a closed buffer system, it is however of minor importance in the ECF. Phosphate contributes substantially to intracellular pH buffering. As intracellular concentrations of phosphate are higher than that in the plasma, phosphate-containing molecules in solution such as DNA, RNA, ATP, and phosphate ions act as buffers. Intracellular pH is more acidic and closer to the pKa of phosphate buffers. Phosphate is also an important buffer in urine. During prolonged acidosis, calcium phosphate in the bone becomes more soluble with prolonged acidosis. Plasma phosphate concentrations then increase, and calcium phosphate can be regarded as an alkali reserve in response to a prolonged acidosis.